Well, I want to thank you for the opportunity to present our group's recent work at PacBio's ASHG workshop. Today, my name is Emily Farrow, and today I'll be discussing an integrated rare disease genomics using long-read genome sequencing. The Genomic Medicine Center at Children's Mercy Hospital was one of the first genome centers wholly embedded within a pediatric setting, and this was largely due to the recognition that rare disease is collectively not all that rare. In fact, there are over 7,700 genetic or inherited diseases. They affect 400 million people globally, which translates to 1 in 10 Americans, half of which are children. Molecular diagnostic yields by symptom-driven clinical exome sequencing in patients with rare disease only range from 30 to 40% in unbiased cohorts. And we now know that the addition of short-read genome sequencing only incrementally increases the diagnostic yield. One of the many advances in rare disease is the discovery of novel disorders and causative genes. In addition to the exponential increase in gene disease association, there is an increased recognition of the complexity of the human genome. Single nucleotide variants, while accurately and efficiently assessed with short-read genome and exome sequencing, are really only one component of molecular diagnostics. We now know that copy number variation, triplet repeat disorders such as Fragile X syndrome, methylation and imprinting disorders such as Prader-Willi or Angelman syndrome, large structural variations such as translocations that appear to be balanced but perhaps aren't, and mitochondrial variants are all contributing to genetic disease. So why are long reads increasingly being utilized and investigated in the context of rare disease? And that's because despite comprehensive sequencing using standard sequencing technologies and integrated informatic approaches, there are multiple remaining analyses challenges. While trio sequencing is powerful, parents are often not available for sequencing. And while this obviously limits the detection of de novo variants, which is critically important for neurodevelopmental diseases, it also is important for autosomal recessive disease, which still accounts for about 21% of cases. Even coverage remains a challenge, even in the face of PCR-free genome sequencing. Repetitive regions such as pseudogenes or expansion disorders, again, such as Fragile X, are difficult to align with short-read technology. And while there have been improvements in structural variant detection, there's still room for opportunities and improvement, particularly in the face of detecting inversions and translocations. And what this amounts to is that when faced with a patient with rare disease, we often are deploying multiple clinical evaluations with multiple assays. And collectively, these can be costly, they can be time-consuming, and they're limited to known, so we can only look at what we know. And in fact, an assay donut hole remains, where one technology can't detect all of the sequencing or copy number changes that the next technology would pick up. Historically, long-read sequencing has been difficult to deploy because of higher error rates and lower throughput. But as these have rapidly been improved, these are now an attractive solution to look at for rare disease. So in rare disease, when assessing for non-methylation, as I mentioned, it changes, laboratories are now utilizing a number of clinical testing methods, ranging from a karyotype to microarray sequencing, expansion, or even inversion testing. In some cases, differentiating which test is most appropriate is completely straightforward, such as suspected trisomy 21, you would start with a karyotype. However, in many cases, such as the case of neonatal hypotonia, testing algorithms are much more complicated. And again, what this amounts to is that with multiple testing assays, we have increased time and increased money. And if we look at average turnaround times for different technologies or assays that are used, we can have 4 weeks for a karyotype. You may have another 4 weeks for a microarray. Depending on which sequencing you're pursuing, it could be 8 to 12 weeks. If you're now suspecting an expansion disorder, such as myotonic dystrophy and hypotonia, that might be another 4 weeks, and so on and so forth. Now, the addition of copy number variant detection from short-read exome and genome sequencing does allow for some testing to begin to be condensed. However, again, these testing cannot detect expansion disorders. Informatic algorithms for PCR-free genome sequencing can screen for known expansions. However, these are still a screening mechanism that require orthogonal confirmation. Improvements in third-generation sequencing, such as the PacBio HiFi sequencing, offer the potential to capture complex copy number variants single nucleotide variants, expansions, and inversions, simplifying our testing algorithms and offering the potential for an increase in diagnostic rates. So in recognition of the fact that over 50% of our patients who receive comprehensive sequencing, including exome sequencing, will not receive a diagnosis, the Children's Mercy Research Institute and the Genomic Medicine Center have created a novel research program called Genomic Answers for Kids. And under this program, we've created a pediatric data repository to facilitate the search for answers and novel treatments for pediatric genetic conditions. 30,000 children and their families will be enrolled over the next 7 years, and comprehensive sequencing and analytic strategies are deployed across the program. As part of our sequencing strategy currently, we now have a systematic reflex to HiFi genomes after a negative exome analysis has been completed. As you may imagine, to support the needed throughput for this program, we spent quite a bit of time optimizing our laboratory preparation methods and our analytical methods. As part of that, our DNA isolation has been completed using a Chemagic 360 automation, which allows for 24 to 48, all the way up to 96 samples to be isolated at once. Samples are sheared to 15,000 base pairs with an input of 5 micrograms of DNA. And library preparation is automated now with the Cyclone G3, which again allows for parallel processing of up to 96 samples at once. In our informatic or bioinformatic algorithms, we also have developed quite a few different strategies. Our sequencing alignment is completed using build 38 for all of our HiFi genomes. Our variant detection is completed using different algorithms, including deep variant, which is used for single nucleotide variants and insertions and deletions. WTAP is used for the phasing of the deep variant calls, and PBSV, or PacBiostructural variants, is used to detect structural variation. For variant annotation, a novel toolset is used to annotate structural variants for population frequency and the impact on known genes. And our repeat expansions are also called tandem genotypes, are annotated for set loci. If we take a very high-level look at our QC metrics, what we can see with our HiFi genomes is that our single nucleotide variants and insertion deletion calls are highly concordant with short-read whole genome sequencing. In addition, the HiFi sequencing is revealing over 450,000 single nucleotide variants and insertions and deletions, and about 10,000 structural variants per genome that were previously undetected. If we apply a fairly conservative allele frequency filter of 1%, less than 1%, we now have about 150 rare variants per genome or per proband for our analysts to investigate that were not detected using our prior technologies. So while sequencing and analysis are ongoing, our initial analyses are already leading to novel discoveries and advancements, particularly in 3 challenging areas that I'd like to highlight today. An ongoing challenge that we have as analysts is the combined analysis of single nucleotide variants and copy number variants. In this case, CMH001017 and the siblings CMH001018 were referred for global developmental delay, hypotonia, strabismus, and MRIs that are consistent with a mild septo-optic dysplasia. And in fact, what was striking about these siblings is that they had a nearly identical clinical presentation. These families have a diagnostic journey of over 7 years that have elapsed since the first test has been ordered. And it's been quite comprehensive, with single gene analyses ordered, deletion and duplications of those single genes, a panel analysis, exome sequencing, and microarray, all of which were non-diagnostic. And then in this case, the family history was non-contributory, and we only have the 2 affected siblings for analysis. We completed HiFi sequencing for both the siblings and the parents. And what we found when this was completed was that they are compound heterozygous for 2 pathogenic variants and a gene AARS2. The first is a missense variant of C to T at position 595, which ultimately results in the replacement of an arginine with cysteine at codon 199, and this was maternally inherited. The second is actually a small deletion at 6p21.1, which includes the deletion of exons 5 through 7 in AARS2, and this was paternally inherited. Now, the deletion is a recurrent deletion that's actually previously been reported, but again, it's falling in this donut hole, whereby it's too small for conventional microarray, yet large enough to be challenging to detect with copy number from clinical exome. AARS2 is associated with a progressive leukoencephalopathy with ovarian failure, and in fact, the full symptoms of this syndrome fit quite nicely with our family. Another aspect that's been optimized is software solutions that are providing us visualization of the variants, allowing for efficient filtering strategies, pedigree analyses, and phenotypic-driven analyses. In this view, you can clearly see the loss of exons 5 through 7, denoted by the large red bar, with a single nucleotide variant with the small caret arrow that have only been seen in this family. Now, again, in this case, we had parental samples, but the power of phasing is evident as it is clear the deletion and pathogenic single nucleotide variant are in opposite alleles. So on the top, you can see the deletion of exons 5 through 7, and then on the right in the red bar, you can see on the second allele the single nucleotide variant. So again, this is consistent with autosomal recessive disease as the variants are in trans. So what are the challenges in this case? Two of them are that the deletions of exons 5 through 7 was not detected by microarray or exome sequencing. So again, it fell in that intermediate area that's difficult to detect. And the second challenge that's agnostic to technology, but an ongoing problem with rare disease, is that AARS2 related disease was actually reported in literature 2 years after the onset of symptoms in the proband and after the last clinical analyses, and not included even on the panel that was originally ordered. And so this again highlights the difficulties we have in keeping up with the new disease gene associations. In keeping with the theme of intermediate copy number changes that are difficult to detect, we had an affected duo that was also referred to our study due to multiple concerns, including growth deficiency, seizures, mild hearing loss, agenesis of the corpus callosum, mild white volume matter loss, severe scoliosis, global developmental delay, and dysmorphic features. And then in this case, the mother, CMH 001061, had a very similar phenotype. The previous testing had identified a deletion at 16q24.3 that was not detected in the daughter when that array was completed. A comprehensive sequencing panel completed on the daughter, or CMH 001060, was also negative. And after a negative exome analysis in our research program, we also completed HiFi genomes on the patient and her mother. And in fact, analysis of the HiFi genomes revealed that both the mom and the daughter carry the deletion at 16q24.3 that had not been reported by microarray in the child. So we do not have access to the original array data, so we cannot speculate on why the call was missed. However, this deletion encompasses exons 1 through 12 out of 13 in a gene ANKRD11, which is consistent with the diagnosis of KBG syndrome. And this syndrome is characterized by short stature, microcephaly, vertebral malformations, seizures in some patients, and developmental delay. The mother had had a diagnostic odyssey of over 30 years, and the probands had been over a decade. And again, this case highlights that intermediate deletions and duplications are still difficult to detect, particularly with older technologies with reduced resolution compared to the new third-generation sequencing. I think combine these two cases together really highlight, again, not only the difficulties in detecting these variants that we're now able to see with the deployment of HiFi genomes, but also the increasing recognition of small copy number variants and their role in genetic or rare disease. Building on our structural variant detection, another class of diseases are expansion disorders. Now, many classic genetic diseases are attributed to repeat expansion disorders, such as Fragile X, myotonic dystrophy, and Friedreich ataxia. Most commonly now, each test is completed independently using repeat primed PCR, replacing the traditional Southern blotting. And despite this huge advance, in many cases, the appropriate assay is still not completed, leading to the under-ascertainment of disease. And this is particularly true, we think, in Friedreich ataxia. Additionally, both the repeat number and the sequence and motif of the repeat are important. We now know that interruptions can modify disease severity in Fragile X, for example. So informatic algorithms for short reads are able to estimate expansions at known loci for PCR-free genomes, and they do actually well in their estimates. However, these algorithms don't give the full sequence or the interruptions. And so now in our program, each HiFi genome is analyzed for repeat expansions with a tandem genotype output that allows for quick scanning of repeat numbers. And then the expansion can also then be directly visualized using IGV. So in this case, CMH003372-04 was diagnosed clinically with Fragile X due to developmental delay. And this has been detected using the triplet prime PCR. However, the expansion number and the potential for sequence interruptions was unknown, as the report states fully expanded consistent with disease. So as a positive control to develop the tandem genotyping tool, a HiFi genome was completed, revealing an expansion of almost 200 repeats. As you can see in the top panel, the one allele, an average or predominant allele is 200, which is consistent with the male with a single X and Fragile X disease. However, you also can see that there are some alleles with a larger expansion, which is not unexpected in a fully expanded allele with Fragile X disease. And in fact, when we look at within IGV closer, we can see that most of our alleles are having a 600 base pair, which would be a 200 repeat, triplet repeat expansion called. And we do in this view see a single expansion of 400 repeats, consistent again, both with our tandem genotyping tool and with what had been seen clinically. So we're particularly excited to deploy the tandem genotyping tool as expansion disorders are particularly common in neuromuscular disease, which is a common referral within our center. Again, currently, repeat expansions are graphed for 51 loci for each sample. And this has resulted in the carrier identification for Friedreich's ataxia and in females that have one allele consistent with Fragile X or expanded allele. Nicely, loci can be added and removed to the tool as the literature may report new expansions or as we've discovered novel expansions. And it's important to remember that the tandem genotyping tool gives us a quick way to screen for expansions, but the insertions are also called by the informatic pipeline, which has also allowed for the novel gene discovery of an expansion for a family we previously presented on STAR-D7. So although the sequencing of expansions is far improved with long reads, there are still some considerations and opportunities for development. Extreme expansions may be difficult to align even in the face of 12,000 base pair reads and may lead to reduced coverage to be aware of. Some specific repeat motifs are difficult to sequence, such as GAA for Friedreich ataxia. And in this case, what we can do is we can do a manual inspection of all the reads, and that can be very helpful in resolving what the expansion and what the repeat insertion is. And we've been quite successful in this regard. Mitochondrial disease is another often underappreciated disease mechanism. It affects nearly 1 in 5,000 births, and onset ranges from more severe presentations in childhood to milder presentations in adulthood. Symptoms are wide-ranging and can include myopathy, lactic acidosis, seizures, and ataxia in childhood, to again milder presentations such as hearing loss and migraines in adults. The mitochondrial genome itself is only 16,569 base pairs with 37 genes. In contrast, the nuclear genome encodes over 1,000 mitochondrial genes important for mitochondrial function. And despite the difference, pathogenic variants in the mitochondrial genome, including a recurrent 5,000 base pair deletion, account for just a little under 15% of mitochondrial-related disease. Therefore, particularly in rare disease, evaluating the mitochondrial genome is important. With sample preparation modifications in our hands, the HiFi genome now captures the mitochondrial genome with an average coverage of 100 to 200x per sample. So again, using the current automated sample preparation techniques that we have, we've preserved the mitochondrial genome, allowing for a complete analysis. And in some cases, the entire mitochondrial genome is covered in a single HiFi read, providing the ability for accurate copy number variant detection, again, a common disease mechanism. Further, our integrated software solutions also include the analysis and visualization of the mitochondrial genome. The one caveat is that in some cases, low level of heteroplasmy, which would look like a low variant allele fraction, in this case would not be detected if it's below 1% or very low in our samples. So if we look at the progress we've made with our HiFi genomes, we've now completed over 600 HiFi genomes to date and still counting. Again, the sequencing and analyses are ongoing. And in our diagnostic findings, we found multiple copy number variant deletions and duplications that are attributed to disease. We've discovered single nucleotide variants, and we've also found repeat expansion disorders that were not found previously. The only thing is we're essentially missing methylation currently with our techniques, which is still important for imprinting disorders, and at this point would still require reflex testing. However, we've shown that what now may take 6 tests can potentially be done with 2 tests, which is a significant improvement. So in summary, what we've shown is that we've been able to automate the sample preparation to support the large-scale deployment of HiFi sequencing in a rare disease program. The comprehensive integrated analyses allow for the simultaneous detection of multiple types of genomic variation, providing cost and time savings. And ultimately, the systematic implementation of HiFi genomes continues to lead to new diagnoses previously unidentified with standard technologies. And I would really like to take a moment to acknowledge our team, particularly our director, Dr. Tomi Pastanen, and all the coworkers, both within Children's Mercy and within PacBio that we've worked with in collaboration. And most importantly, I'd really like to acknowledge the patients and their families for their participation in this report, because without them, we wouldn't have a program. And with that, I thank you for your time.